Good morning. So you learned about glycogenolysis and glycogenesis. What is glycogenolysis? Breakdown of glycogen to form glucose. Why should glycogen be formed and why should it be broken down? You've seen that glucose following meal is stored as glycogen for use in between the meals. You learned that for 2 to 3 hours after the food, glucose is available by absorption. After that, glucose will not be available. And then we have seen that brain erythrocytes, particularly, they require glucose all the time. And you've seen for some other purpose, for citric acid cycle to be maintained and for synthesis of lactose in lactating mothers, glucose must be available. So that's why glycogenolysis provides the glucose necessary during in between meals in the post-absorptive state. So the first step in glycogenolysis involves glucokinase. What is this glucokinase? In which tissue it is present? It is present only in liver, and it is not inhibited by its product, glucose 6-phosphate, and its Km value is high, so that's why it's only at a high glucose concentration it will be active. So following meal, immediately following meal, blood glucose level is quite high, and so glucokinase is active, and it is not subject to, it is not inhibited by feedback inhibition. So these are the advantages of glucokinase. So with the action of glucokinase, the liver converts glucose to glucose 6-phosphate, whereas in the muscle, hexokinase only, there is no other alternative there. So in the muscle, it's hexokinase, whereas in the liver, the special enzyme glucokinase converts glucose to glucose 6-phosphate for glycogenesis. And then subsequently you have seen that 4 more steps will be required. So this is glucokinase converts it to glucose 6-phosphate, then phosphoglucomutase or phosphohexose mutase converts glucose 6-phosphate to glucose 1-phosphate, which in the presence of UDP pyrophosphorylase, this enzyme requires UTP, and in the presence of UTP, it forms UDP glucose, removing inorganic pyrophosphate, and pyrophosphatase hydrolyzes it to 2 molecules of inorganic phosphate, pulling this reaction from glucose 1-phosphate to formation of UDP glucose. And this UDP glucose, it is in this form. It is the active glucose residue that can be added to a glycogen primer or to an existing glycogen molecule. So in the presence of an existing glycogen molecule, 1 glucose residue is added. So 1 glucose residue is added at the non-reducing outer end. So if this is the glucose to be added, the UDP glucose, it comes, and then UDP is removed and glucose residue is added to the C4 here, forming a glycosidic bond. Now, in the first time when there is no primer, from a protein called glycogenin, which contains tyrosine residues, it is formed, and one of these specific tyrosine residue takes up the glucose from UDP glucose, and then 7 more glucose residues are added, which forms the glycogen primer. So to that UDP glucose, 1 glucose molecule is added by UDP glucose each time, until there are about 11 residues in a chain. So when there are about 11 residues, say here, branching enzyme acts and it removes a 6 glucose residue piece to an existing branch, forming a branch point with alpha 1,6 glycosidic branch. So here it is alpha 1,4 glycosidic bonds, whereas at the branch point it is alpha 1,6 glycosidic bond. See this? This is the 4,1, whereas here it is 6,1. At the branch point it is alpha 1,6 glycosidic linkage. So then elongation of this again takes place and the process is repeated. So that is about glycogenesis. And you have seen that the rate limiting enzyme of glycogenesis is glycogen synthase. Whereas glycogenolysis, the reaction first starts with the rate limiting enzyme, that is glycogen phosphorylase or phosphorylase, which is the rate limiting enzyme of glycogenolysis. It causes phosphorolytic breakdown. So that is inorganic phosphate it takes from the cytoplasm, and then the enzyme glycogen phosphorylase cleaves off 1 glucose residue to form glucose 1-phosphate. So that is glycogen phosphorylase, the rate limiting enzyme. Now, this goes on. So Glucose 1-phosphate is then acted upon by phosphoglucometase to form glucose 6-phosphate, and then it is acted upon by glucose 6-phosphatase to form glucose, which is released by the liver into the blood to be utilized by the other tissues. Whereas in the muscle, there is no glucose 6-phosphatase, and so glucose 6-phosphate is utilized only by the muscle. And then when there are about 4 residues, now phosphorylase cannot act. So now a debranching enzyme, which consists of 2 parts. One is alpha 1,4 to 1,4 glucan transferase and alpha 1,6 glucosidase. So this alpha 1,4 to 1,4 glucan transferase transfers a trisaccharide unit. So from here to an existing branch. So it is transferred here, exposing the branch point, which is then acted upon by alpha 1,6 glucosidase to form free glucose. Here, while phosphorylase, you have seen that it releases glucose 1-phosphate. This debranching enzyme releases free glucose. That's what you should remember. So while phosphorylase releases glucose 1-phosphate, debranching enzyme releases free glucose residues. That you should remember. So regulation of hepatic glycogenolysis. So glycogenolysis is activated in the liver in the fasting state, 1A1. And the 3 major hormones activate hepatic glycogenolysis. So it is not only in the fasting state, not only the post-absorptive state, but following acute stress and chronic stress also it is released. And let us see how it is regulated. So 3 major hormones activate this hepatic glycogenolysis: glucagon, epinephrine, and cortisol. So glycogenolysis is brought about in the post-absorptive state by glucagon. Acute stress causes an activation of glycogenolysis through the action of epinephrine. So epinephrine causes glycogenolysis in acute stress. Whereas during prolonged exercise, both epinephrine and glucagon activate glycogenolysis. Whereas under continuously stressful conditions, such as psychological stress or cold stress, cortisol brings about glycogenolysis. So this is what you should remember. So in the post-absorptive state, it is by glucagon. In the acute stress, glycogenolysis is brought about by epinephrine. During prolonged exercise, both epinephrine and glucagon activate glycogenolysis to maintain blood glucose level. Whereas under continuously stressful conditions, cortisol increases or regulates or brings about glycogenolysis. What is the mechanism of their action? Glucagon activates hepatic phosphorylase through activation of adenyl cyclase. Whereas cortisol, acting at the genetic level, binds to hormone responsive element and induces transcription of the genes of hepatic phosphorylase. Whereas epinephrine acts through alpha and beta adrenergic receptors, which you have seen. Okay, I will explain once again. So this is the action of cortisol, glucocorticoid response element or hormone response element. It goes and binds. So this hormone goes through the cytosol and through the nuclear membrane and it binds to nuclear receptor protein at the hormone response element to the DNA. Then it increases the transcription of mRNA and then protein synthesis. Whereas glucagon particularly, so when it has got a receptor at the cell membrane, so cell membrane surface receptor, it stimulates, when it binds, it activates a G protein. The G protein consists of alpha, beta, gamma. It's a trimer, and to the alpha, GDP is attached. And when hormone binds, it is activated whereby GDP is replaced by GTP, and alpha GTP binds to the enzyme on which it acts, releasing beta gamma dimer. So now this enzyme, adenyl cyclase, is activated. So there is amplification at each of the following steps. So here, one adenyl cyclic AMP molecule that is formed activates several G protein molecules and so on. So this adenyl cyclase forms cyclic AMP from ATP, and this cyclic AMP activates inactive protein kinase. It consists of R2C2, regulatory and catalytic subunits, 2 regulatory and 2 catalytic subunits. So when the cyclic AMP binds, these regulatory subunits are released from it. So these are the inhibitory one that will be inhibiting, holding it on. So keeping it in an inactive state. So as the R2 is removed by this cyclic AMP. So now see, it becomes active protein kinase A. And this protein kinase A then activates further enzymes. So cyclic AMP activates protein kinase A. Protein kinase A activates I'll show you. Next slide. So cyclic AMP dependent protein kinase is activated, which activates phosphorylase kinase B by phosphorylation from ATP, utilizing ATP, and phosphorylase kinase activates phosphorylase B to phosphorylase. So this is the dephosphorylated form, this is the phosphorylated form. So as the phosphorylase is phosphorylated by phosphorylase kinase, it becomes active. So phosphorylase B is in active form, it is activated to phosphorylase A. And then insulin activates an enzyme called phosphatase, protein phosphatase. So it also increases glucose 6-phosphate and activates phosphatase. So glucose 6-phosphate as such inhibits phosphorylase B. So you can see that it is inhibited. Conversion of phosphorylase B to A is inhibited. Whereas protein phosphatase dephosphorylates this phosphorylase kinase A to form inactive phosphorylase kinase. And this protein phosphatase is inhibited by an inhibitor 1, which is activated by cyclic AMP. So these steps you should remember. Insulin also activates an enzyme called phosphodiesterase. So this phosphodiesterase converts or hydrolyzes cyclic AMP to 5' AMP. So the whole cascade is blocked now. So that is one. And apart from that, insulin also activates protein phosphatase that you have seen, which dephosphorylates, thereby inactivating active phosphorylase. Then inhibitor 1 activated by cyclic AMP dependent protein kinase inactivates protein phosphatase, facilitating glycogenolysis. That's what you have seen. Caffeine in coffee and theophylline in tea are also inhibitors of phosphodiesterase and thus enhance hepatic cyclic AMP and increases glucose supply to the blood. And that's why you get a stimulating effect when you take coffee. You have more cups, not just probably one. But thus, while glucagon brings about hepatic glycogenolysis, insulin inhibits glycogenolysis. Active phosphorylase A in both the tissues is inhibited by ATP and glucose 6-phosphate. So you've seen that glucose 6-phosphate. So ATP also inhibits phosphorylase B conversion to phosphorylase A. In liver, free glucose also inhibits glycogen phosphorylase. So when there is more glucose, there is no need of glycogenolysis. So mechanism of action of epinephrine, it is slightly different. So this epinephrine works through its alpha and beta adrenergic receptors. During severe hypoglycemia, glucagon and epinephrine you have seen work together to increase hepatic glycogenolysis. In response to real or perceived threats, even when blood glucose is normal, epinephrine is released, enhancing glycogenolysis and thus raising blood glucose level to support a fight or flight response, which you should remember. So this epinephrine acts on hepatic glycogenolysis by two pathways. It consists of beta adrenergic receptors. So action of epinephrine through the beta adrenergic receptors is similar to that of glucagon acting through cyclic AMP, adenyl cyclase, cyclic AMP dependent protein kinase, phosphorylase kinase, and phosphorylase. Whereas binding to alpha receptors, epinephrine activates G protein that activates phospholipase C, hydrolyzes phosphatidyl inositol. So hydrolyzed phosphatidyl inositol, and this diphosphate protein phosphorylase. AMP independent phosphorylation. That's what you should remember. The cyclic AMP does not have a role in this inositol triphosphate or phospholipase C action or phosphatidyl inositol biphosphate action. But glycogenolysis in muscle is slightly different. In muscles, 5' AMP acts as an allosteric activator of dephosphorylated B form of phosphorylase. Muscle contraction increases intracellular calcium concentration. So as muscle contracts, intracellular calcium concentration will increase. This increases glycogenolysis several fold. So calcium ions bind to calmodulin, activating calmodulin sensitive phosphorylase kinase. So this muscle phosphorylase kinase is a tetramer made of alpha, beta, gamma, and delta subunits. Alpha and beta subunits are phosphorylated by cyclic AMP dependent protein kinase. So beta. Whereas alpha, the delta subunit binds calcium ions, which activates gamma subunit, while the enzyme is in dephosphorylated form itself. So this gamma, it is acted upon. Action here is on this delta subunit, which activates gamma subunit and then activates phosphorylase kinase. So when this fright or fight, epinephrine secretion causes cyclic AMP formation and glycogenolysis. Similarly, regulation of glycogenesis also will learn. Glycogen synthase also exists in phosphorylated and dephosphorylated form, but here it is reversed. Phosphorylated form is inactive, while dephosphorylated form is active. So both calcium, calmodulin dependent protein kinase and cyclic AMP dependent protein kinases, they phosphorylate and inactivate glycogen synthase. So this is brought about synchronously with activation of phosphorylase and so glycogenolysis. So when phosphorylase is activated, glycogen synthase is inhibited, and when glycogen synthase is activated, phosphorylase is inhibited. So synchronously or simultaneously. So see this? So glycogen synthase, calmodulin dependent protein kinase inactivates glycogen. So phosphatase is activated by insulin and glucose 6-phosphate. And this protein phosphatase activates glycogen synthase A, glycogen synthase B to glycogen synthase A, which brings about glycogenolysis. So here cyclic AMP dependent protein kinase or phosphorylase kinase or calmodulin dependent protein kinase, they convert the active form of glycogen synthase to inactive form while activate phosphorylase. You should remember that. So glycogen synthase and phosphorylase are thus reciprocally regulated, are simultaneously regulated. So reciprocally, opposite action, opposite to each other. So insulin secreted in response to a high blood glucose concentration activates glycogen synthase and inhibits phosphorylase by raising the concentration of glucose 6-phosphate, which stimulates phosphatase, causing activation of glycogen synthase. So this you should remember. Insulin secreted in response to a high blood glucose concentration activates glycogen synthase. How? By activating glucokinase and so forming English, it's a glucose 6-phosphate. This inhibits phosphorylase. So this stimulates phosphatase, causing activation of glycogen synthase. Insulin also activates phosphodiesterase, so cyclic AMP is hydrolyzed, so that cascade is blocked. So when phosphorylase is activated by rise in cyclic AMP concentration, by glucagon or by epinephrine, glycogen synthase is inactivated. Thus, glycogen metabolism is regulated reciprocally by glycogen synthase and phosphorylase. So I had just introduced glycogen storage disorders for you. So of which, from the examination point of view, it is Van Gierke's disease which is important, but from the patient point of view, type 2 and 4 are important because they are fatal. So this type 1 disorder or Van Gierke's disease is caused by the deficiency of glucose 6-phosphatase in liver. Glucose 6-phosphate accumulates as a result. So it cannot leave into the blood from the liver cell. The blood glucose level, but blood glucose level is low. Glucose phosphate in liver is high, but blood glucose level is low. So when the energy content of the cells is low, it stimulates lipolysis and then ketosis to provide alternate source of energy. So apart from that, this elevated hepatic glucose 6-phosphate levels increase pentose phosphate, resulting in increased synthesis and catabolism of purine nucleotides, resulting in hyperuricemia. So purine nucleotide catabolism leads to uric acid production. So there is increased purine nucleotides, increased catabolism, so hyperuricemia. So elevated glucose 6-phosphate levels also increase its conversion to glycerol and fatty acids, and so increase serum triglyceride. Whereas type 2 or Pompey's disease is due to deficiency of lysosomal 1,4-1,6 glucosidase or branching enzyme. So here branching cannot take place. Glucose without much branching so occupies much space in the cell, resulting in accumulation of abnormal glycogen in all tissues, particularly in heart, resulting in heart failure. So glycogen molecule occupies the whole heart, expands it, so resulting in heart failure and death by ACE2. That's how this type 2 import is fatal. So type 3, limit dextrosis or Cory's disease is due to deficiency of debranching enzyme. Resulting in here, it is lysosomal branching enzyme. That's what you should remember, type 2. Here, it is deficiency of debranching enzyme, resulting in accumulation of abnormal glycogen in liver, leading to hypoglycemia and hepatomegaly. Type 4 Anderson's disease or amylopectinosis is due to deficiency of branching enzyme in liver and muscle, so resulting in accumulation of abnormal glycogen, leading to hepatosplenomegaly and death due to hepatic or heart failure in first year of life. And that's how type 2 and type 4 are fatal. Type 4 is the McArdle's disease or McArdle syndrome is due to muscle phosphorylase deficiency. And so accumulation of muscle glycogen takes place. There will be poor exercise tolerance and lactic acidosis. Type 6 is the hearse disease due to deficiency of hepatic phosphorylase, accumulation of glycogen in liver, hepatomegaly, hypoglycemia. Whereas Torres disease is due to deficiency of phosphofructokinase in muscle and erythrocyte. And you see, it is the rate limiting enzyme of glycolysis. When the glycolysis cannot take place, there will be hemolytic anemia. Erythrocytes will be destroyed, and so there will be hemolytic anemia apart from poor exercise tolerance and accumulation of glycogen in muscle. So that is about glycogen metabolism. Then coming to gluconeogenesis. So this gluconeogenesis means synthesis of glucose from non-carbohydrate precursors. So in the post-absorptive state, in the fasting state, as the glycogen comes to be almost hepatic glycogen stores start getting depleted, and then gluconeogenesis provides the glucose necessary for the tissues. So it takes place liver mainly and kidney to lesser extent. Whereas glycogenesis is seen takes place in liver and muscle. Gluconeogenesis takes place in liver and kidney. So this is partly mitochondrial and mainly cytosolic. So cytosolic is same for both the glycogenesis, glycogen metabolism, and for gluconeogenesis, but gluconeogenesis is partly mitochondrial. That's what you should remember. So what is the importance of this gluconeogenesis? Why should the non-carbohydrate precursors should be converted to glucose? Even in fasting and starvation, when fatty acids are the main source of energy in the tissues, blood glucose must be maintained above 60 mg/% so that brain and RBC get a continuous supply of glucose. Otherwise, it goes to hypoglycemia, causes brain dysfunction, coma, and death. You have seen that. And to maintain the levels of intermediates of the citric acid cycle and for the synthesis of lactose. Apart from this, gluconeogenesis has an important function, a scavenging function. Scavenger is the one that removes the waste products. So here are the waste products that are lactate produced by muscle and erythrocyte during glycolysis, and glycerol and propionate produced by adipose tissue during lipolysis are removed by this gluconeogenesis. Of course, proteolysis in the fasting state or starvation also provides many glucogenic amino acids, which are substrates, which are the main substrates for gluconeogenesis. So gluconeogenesis becomes the primary source of blood glucose after about 8 hours of post-absorptive period, that is the starvation. So gluconeogenesis requires a source of energy for biosynthesis and a source of carbons for the formation of glucose. So that's what you should remember. So gluconeogenesis, you have seen the sources of carbon are the lactate, glycerol, propionate, and glucogenic amino acids, whereas the energy source is by oxidation of fatty acids. So oxidation of fatty acids released from adipose tissue provides the energy, while the carbon sources are lactate, glycerol, propionic acid, and glucogenic amino acids. So the lactate is produced during glycolysis by RBC and under anaerobic conditions by muscle via pyruvate. Glycerol is produced by adipose tissue during lipolysis. Propionic acid is produced by the metabolism of fatty acids with the odd number of carbon atoms, by the side catabolism of side chain of cholesterol and of isoleucine and methionine. Glucogenic amino acids, particularly alanine, produced by protein breakdown. Proteolysis also provides alternate source of energy in the starvation. So, among the above, the muscle protein is the major precursor. During prolonged fasting, starvation, or malnutrition, both adipose tissue and muscle mass, that's where they are lost. Both the meltdown, they are hydrolyzed, then catabolized. So, the fat is used for the general energy needs of the body and also for gluconeogenesis, while most of the amino acids in the protein are converted into glucose. So, only a small amount of energy will be provided by these amino acids, and mainly for utilized for gluconeogenesis. So now seeing each of these substrates, how they're converted to glucose. So you've seen lactate. Lactate produced in muscle and erythrocytes are converted to pyruvate by lactate dehydrogenase. So lactate, NAD, the CHOH is oxidized to CO, and this NAD takes the hydrogen, the two hydrogens formed pyruvate. So gluconeogenesis. So you should remember this difference between lactate and pyruvate. They're interconverted by lactate dehydrogenase, whereas the action of pyruvate dehydrogenase is only on pyruvate, convert it into acetyl-CoA. This you should not forget. So gluconeogenesis from pyruvate is mainly reversal of glycolysis. So it is now pyruvate that is to be converted to glucose. So it is conversion of pyruvate to glucose is reversal of glycolysis. So you have seen that it is conversion of pyruvate to glucose, which has to be reversal of glycolysis. But you learned there are 3 reversible steps in glycolysis, catalyzed by one by hexokinase, 2 phosphofructokinase, and 3 pyruvate kinase. These are the 3 irreversible enzymes which are to be catalyzed by alternative enzymes. And there is also a fourth one which catalyzed by phosphoglycerate kinase to convert 3-phosphoglycerate to 1,3-diphosphoglycerate. So 1,3-diphosphoglycerate to 3-phosphoglycerate is easy. By hydrolysis, ATP is hydrolyzed. But here ATP is added, which is an active step, which doesn't take place just like that. So it requires ATP and it requires so it is another step. So 4 irreversible steps, in fact. 4 irreversible steps. So but these 4 irreversible steps, to overcome these 3 irreversible reactions, liver utilizes 4 enzymes. So apart from the phosphoglycerate kinase, which acts in a reverse direction, utilizing ATP. So this pyruvate carboxylase, phosphoenol pyruvate carboxykinase, fructose 1,6-biphosphatase, glucose 6-phosphatase. These are the 4 irreversible enzymes to overcome the 3 irreversible reactions. So what are the 3 irreversible steps of glycolysis? So you have seen that it is hexokinase, phosphofructokinase, and pyruvate kinase. Pyruvate kinase is in the next slide. So pyruvate kinase. These are the 3 irreversible enzymes of glycolysis are to be overcome. So this is done by 4 enzymes, that is pyruvate carboxylase, phosphoenol pyruvate carboxykinase, fructose 1,6-biphosphatase, and glucose 6-phosphatase. So pyruvate carboxylase, this is present in mitochondria. This is a mitochondrial enzyme, whereas pyruvate is formed in glycolysis in that takes place in cytosol. So first this pyruvate carboxylase, see its action. So in the presence of ATP and carbon dioxide, this enzyme requires biotin as a cofactor. So in the presence of that, pyruvate is converted to oxaloacetate. But since it is in mitochondria and pyruvate cannot cross, so the oxaloacetate which is in mitochondria is to be converted. Oxaloacetate which is in mitochondria is to be converted to phosphoenol pyruvate by phosphoenol pyruvate carboxykinase, which is cytosolic. So mitochondria are not permeable to oxaloacetate. Okay. It is not this step. So pyruvate, by means of a transporter, can pass through into the pyruvate readily. So now it is pyruvate is acted upon by pyruvate carboxylase, which is mitochondrial enzyme, and now oxaloacetate is found. But this oxaloacetate cannot come out of the mitochondria. So it is acted upon by, so this oxaloacetate is converted first to malate by a reversal of malate dehydrogenase action, and this malate can come out of the mitochondria into the cytosol, and this malate is then first acted upon by once again malate dehydrogenase in the cytosolic malate dehydrogenase, forming oxaloacetate, and this oxaloacetate is acted upon by the next irreversible enzyme of gluconeogenesis, that is phosphoenol pyruvate carboxykinase. So the cytosolic phosphoenol pyruvate carboxykinase catalyzes decarboxylation. You see this? So the COOH or this COOH is removed. So this CO becomes OH, COOH. So CH2, and this is phosphorylated COP, and then so CH2, this is decarboxylated carbon dioxide goes away, CH2, COP, so this is phosphorylated, and COOH. CH2, COP, COOH, that is phosphoenol pyruvate carbon dioxide is removed, and GTP is hydrolyzed to GDP, and this phosphate is incorporated into this to form phosphoenol pyruvate. So in liver and kidney, the reaction of succinate thiokinase, if you remember the TCA cycle, here in the TCA cycle produces GTP, so it is necessary here rather than ATP as in other tissues. So in kidney and liver, so which are the tissues of gluconeogenic tissues. So the subsequent reactions from phosphoenol pyruvate through 2-phosphoglycerate and 3-phosphoglycerate are reversible. So 3-phosphoglycerate to 1,3-biphosphoglycerate to glyceroldehyde 3-phosphate, 1,3-biphosphoglycerate, 3-phosphoglycerate. So the 3-phosphoglycerate. So phosphoenol pyruvate has then converted to 2-phosphoglycerate to 3-phosphoglycerate. So those steps are reversible. Now this 3-phosphoglycerate, it can't be just converted to 1,3-diphosphate. Just it's not, though it is an enzyme, same enzyme, but here ATP is to be provided. So that's what you should remember. So then glyceroldehyde 3-phosphate is formed. So 3-phosphoglycerate to 1,3-biphosphoglycerate, 1 ATP is to be spent. Then the steps from 1,3-biphosphoglycerate to fructose 1,6-biphosphate by glyceroldehyde 3-phosphate dehydrogenase, aldolase, and phosphotryptophan, they're reversible. The next irreversible step of glycolysis to be bypassed is conversion of fructose 1,6 biphosphate to fructose 6-phosphate, and it is catalyzed by the gluconeogenic enzyme fructose 1,6 biphosphatase. So while fructose 6-phosphate is converted to fructose 1,6 diphosphate in glycolysis by phosphofructokinase, in gluconeogenesis, fructose 1,6 biphosphate is converted to fructose 6-phosphate by fructose 1,6 biphosphate. Now, the last irreversible step to be overcome is conversion of glucose 6-phosphate to glucose. It is catalyzed by glucose 6-phosphatase. Now, you've seen lactate is the one substrate from which you have seen gluconeogenesis. So it is mainly pyruvate conversion to glucose. The second is you've seen that glucogenic amino acids. Rather, this is the major one, but lactate is produced constantly. That's why we discuss that first. But the major substrate for gluconeogenesis are the glucogenic amino acids. So glucogenic amino acids are formed by proteolysis. So glucogenic amino acids in pyruvate are intermediates of TCA cycle, which can participate in gluconeogenesis. So alanine and glutamine are the major amino acids exported from muscle for gluconeogenesis. Glutamate and alanine, they are the major substrates for gluconeogenesis. So this alanine can be formed from pyruvate. So alanine forms pyruvate rather. So tryptophan forms alanine. So alanine can form pyruvate. Whereas this hydroxyproline, serine, threonine, cysteine, and glycine, they form pyruvate. It is in the TCA cycle, it's easy for you to remember probably. So hydroxyproline, serine, cysteine, threonine, and glycine, they form pyruvate. Whereas tryptophan forms alanine. Alanine can be converted to pyruvate by transamination. Apart from that, you see, glutamate is formed from glutamine, and histidine, proline, and arginine, they all form glutamate, so which on transamination forms alpha-ketoglutarate, the substrate of an intermediate of TCA cycle. Whereas succinyl-CoA is formed from isoleucine, methionine, and valine. Fumarate is formed from phenylalanine and tyrosine, whereas aspartate on transamination forms oxaloacetate. So these are the intermediates of the TCA cycle. So alanine is directly converted to pyruvate by alanine transaminase, glutamate pyruvate transaminase. And then gluconeogenesis takes place as for lactate or pyruvate. It's in hydroxyproline, serine, cysteine, and glycine forming pyruvate. Tryptophan produces alanine. Other glucogenic amino acids produce intermediates of TCA cycle, aspartate, oxaloacetate, glutamine, proline, histidine, and arginine, then metabolized to glutamic acid, which on transamination is alpha-ketoglutarate. Methionine, isoleucine, valine are metabolized to succinyl-CoA, phenylalanine, tyrosine, or metabolized to fumarate. So then there's gluconeogenesis of these amino acids takes place. And you've seen the third substrate, the glycerol, which is produced by lipolysis. In a fasting state, adipose tissue releases glycerol by lipolysis. This glycerol is in gluconeogenesis by liver and kidney. So in the adipose tissue, glycerol kinase that activates glycerol to glycerol phosphate is lacking. So glycerol is transported through the blood to the liver, where it is acted upon by glycerol kinase. See this? Glycerol kinase has to, in the adipose tissue, it is not there. So it's why it is to be brought to the blood. Through the blood, it is brought to the liver. It is acted upon by glycerol kinase to form glycerol phosphate, which forms dihydroxyacetone phosphate with action of glycerol 3-phosphate dehydrogenase. And dihydroxyacetone phosphate is in the glycolysis. NAD-dependent dehydrogenase is converted to dihydroxyacetone phosphate, an intermediate of glycolysis, and gluconeogenesis can be converted by gluconeogenesis, it can be converted to glucose. See this? So glycerol is acted upon by glycerol kinase to form glycerol 3-phosphate. Then it is acted upon by dehydrogenase, NAD-dependent dehydrogenase to form dihydroxyacetone phosphate. Then the steps, in the reversal of glycolysis. Conversion of propionate to glucose, another substrate that is formed from beta oxidation of odd chain fatty acids, metabolism of isoleucine, and oxidation of side chain of cholesterol. These all can produce this propionate. And propionate is to be converted to glucose. So the first step is activation to propionyl-CoA by acyl-CoA synthase or thiokinase. Right? By acyl-CoA synthase or the kinase, acyl kinase, which utilizes ATP and forms AMP. This is what you should remember. Propionate is activated to propionyl-CoA. Propionyl-CoA then to create a carbon, it undergoes carboxylation. So the carboxylases, they all are biotin dependent and require ATP. So propionyl-CoA, by the action of propionyl-CoA carboxylase, is converted to dimethylmalonyl-CoA. So carbon CO is added here. It is carboxylation at the C2 forming D-methyl-malonyl-CoA. Methyl-malonyl-CoA racemase then acts upon it to convert it to L-methyl-malonyl-CoA. And L-methyl-malonyl-CoA, in the presence of the methyl-malonyl-CoA enzyme isomerase, which requires 5-deoxyadenosylcobamide, one coenzyme of vitamin B12, forms succinyl-CoA. So here you can see this. So COOH, which was there on C2, it is now shifted on to COOH. So such isomerization reactions, they require this deoxyadenosylcobamide, and methyl-malonyl-CoA isomerase is the enzyme. So now succinyl-CoA is an intermediate of TCA cycle, and then you can know how it is. So succinyl-CoA is converted to oxaloacetate in the TCA cycle, and then oxaloacetate is converted to glucose. And deficiency of cobalamin can cause methylmalonic aciduria. So it's not converted to, when there is deficiency of vitamin B12, so methylmalonyl cannot be converted to succinyl-CoA, and methylmalonic acid is excreted in urine, that is called methylmalonic aciduria. Energetics of gluconeogenesis. So you learned that 4 irreversible enzymes are the ones that are necessary for gluconeogenesis. What are they? Pyruvate carboxylase, phosphoenol pyruvate carboxykinase, fructose 1,6 biphosphatase, and glucose 6-phosphatase. This you should always remember. See this? The 4 irreversible enzymes of gluconeogenesis: pyruvate carboxylase, phosphoenol pyruvate carboxykinase, fructose 1,6 biphosphatase, and glucose 6-phosphatase. So what are the energetics? How much energy is to be spent for synthesis of 1 molecule of glucose from 2 molecules of pyruvate? So 1 ATP is for pyruvate carboxylase, 1 GTP is for phosphoenol pyruvate carboxykinase, and 1 ATP for phosphoglycerate kinase. So each molecule of pyruvate requires 3 ATPs. So 2 molecules to be converted to glucose requires 6 ATPs spent in the production of glucose from pyruvate. This energy is derived from beta oxidation of fatty acids. Glucose is not there as such to provide energy. It's rare. It is to be saved. It's to be preserved. And so fatty acids are utilized for provision of this energy. Beta oxidation of fatty acids provides the necessary energy. Regulation of gluconeogenesis. So when there is adequate supply of glucose, enzymes of glycolysis become more active, while those of gluconeogenesis have low activity. So they are reversed. So they are reciprocally regulated, enzymes of gluconeogenesis and glycolysis. So when glucose supply is low, enzymes of gluconeogenesis are activated. So that's how dietary regulation. Whereas hormones, insulin inhibits gluconeogenesis by repressing the enzymes of gluconeogenesis. Also, by activating phosphatase, insulin activates pyruvate dehydrogenase. That also promotes glycolysis while inhibiting gluconeogenesis. Insulin inhibits gluconeogenesis. That's what you should remember. Whereas glucagon and glucocorticoids, acting through cyclic AMP, they increase gluconeogenesis. So this cyclic AMP dependent protein kinase is activated, leading to phosphorylation. That is cyclic AMP activates cyclic AMP dependent protein kinase, leading to phosphorylation and inactivation of pyruvate dehydrogenase. So pyruvate dehydrogenase, like glycogen synthase, is inactivated by phosphorylation. So cyclic AMP dependent protein kinase inactivates pyruvate dehydrogenase and accumulation of pyruvate, leading to inhibition of glycolysis. So when the pyruvate accumulates, glycolysis is stopped. Phosphofructokinase is inhibited. So by affecting the concentration of fructose 2,6 biphosphate also, the same effects are brought about. So glucagon and glucocorticoids, they inhibit glycolysis and facilitate gluconeogenesis by affecting the concentration of fructose 6,2,6 biphosphate. What is this fructose 2,6 biphosphate? Let us see. So under a superfluid, that is when there is excess of glucose, phosphofructokinase 2 is activated only in liver. That's what you should remember. While phosphofructokinase 1 is present in all tissues, this phosphofructokinase is present only in liver. It increases the concentration of fructose 2,6 biphosphate. So fructose 6-phosphate, it converts to fructose 2,6 biphosphate, which stimulates glycolysis by activating phosphofructokinase 1. So this fructose 2,6 biphosphate, it relieves the inhibition of phosphofructokinase 1 by ATP and increases its affinity for fructose 6-phosphate. That this fructose 2,6 biphosphate acts. And this inhibits fructose 1,6 biphosphatase and facilitates phosphofructokinase action. So phosphofructokinase 2 is a bifunctional hormone, also having fructose 2,6 biphosphatase activity, not only fructokinase, but also phosphatase activity. So under conditions of glucose shortage, phosphofructokinase 2 activity is low, whereby there is a decrease in the concentration of fructose 2,6 biphosphate and increase in the activity of fructose 2,6 biphosphate. See this? So fructose 6-phosphate. So when there is adequate glucose, fructose 2,6 biphosphate is inactive, phosphofructokinase 2 is active, and that's why it converts from fructose 6-phosphate to fructose 1,6 biphosphate. Whereas when the glucose is in short supply, this phosphofructokinase is inactive, kinase activity is inactive, but phosphofructokinase 2 phosphatase action is active. So this fructose 2,6 biphosphate is converted to fructose 6-phosphate. Fructose 1,6 biphosphate activity is activated. So under superfluity of glucose, phosphofructokinase 2 is activated in liver. It increases concentration of fructose 2,6 biphosphate, stimulates glycolysis by activating phosphofructokinase 1, and inhibits fructose 1,6 biphosphate. Whereas phosphofructokinase 2, it has got fructose 2,6 biphosphate activity, which is activated when glucose concentration is low, and so low, whereby there is a decrease in the concentration of fructose 2,6 biphosphate and increase in the activity of fructose 2,6 biphosphatase, which inactivates phosphofructokinase 1 but activates fructose 1,6 biphosphatase. Okay, we'll continue in the next class.
Thank you, sir.